Mhm. Can I ask meta that you have learned about the 3D culture for me, right?
It was from the
618, right?
yes, correct.
Okay, so okay, thank you. I will try to wrap up quickly because you have already I can that I remember that we have already learned about the 3D um cell culture. Okay, I will start now about the second part, 1 hour about the cancer mimics model, which is the point out about the 3D spheroid and also 3D organoids. So in this lecture, I will show to you why the 3D cell culture is important and point to you about the 3D spheroid and also 3D organoids. And I think this slide that could I ask, have anyone has the experience or had the project or the thesis with the 3D cell culture? There you have? No? Okay. If no, we will go through about the knowledge, the experiment with the 3D cell culture from now. As I mentioned to you that in the cancer research field, one of the biggest challenge is developing the model that mimics the complex behavior in the human body, human tumor. Okay. And you see the traditional model like the 2D cell culture offer the short previous culture result, but it's not fully replicate the full microenvironment and biological interact that is occur in the real tumor in the real patient. One of the most effective approach that refer to use the 3D cancer mimics model. And in my lecture today, I will point to you the 3D spheroid and also 3D organoid, or we call it PDO, patient tissue derived organoid. Okay, 3D cell culture offer the significant improvement because it mimics the natural environment of the cell much more than the 2D cell culture. Also, following this, my lecture, we will go through over the 3D cell culture. It's more detail, but because the miss of time, I will point to you just only to 3D cell, spheroid, and also the organoid. Okay, come again with this literature review is about the 3D model is include when we search on the PubMed that you see is increased a lot about the publication in the y-axis is about the number of publication that use the 3D model in the publication you see until now is quite popular in this field of the cancer research field. And because of the 3D model have become a game changer in the disease research field. One of the key reasons, the key factor that they have the ability to achieve more biological relevance than those that through the traditional cell culture. Okay, they provide like the more biological relevant condition, bring the gap between the cell culture experiment and the real life of the patient response. Okay, and you can see here the 3D structure of the tumor cell. We need the cell-to-cell interaction or ECM to cell interaction also. In the yellow line like this around the tumor mass is the extracellular matrix that give the support for the structure of the 3D of the cancer. And another cell-to-cell, for example, stromal cell, fibroblast, fibrin, or everything, the immune cell also, the 3D structure, the 3D model can demonstrate or mimics the real situation in the real patient. Okay, so show to you again in this picture about the right hand side show the comparison between the 2D and 3D cell culture model. 2D cell culture is mean the cell that can grow in the flat plaster dish or plaster culture ware is grow in only the 2D monolayer. Even proliferation and migration invasion is occur in only in monolayer. Compare with the 3D structure like this, the media and also the component that we put in the culture is support the cell to grow in the 3D structure. Okay, so in this design, we allow the cell grow in 3D that quite mimic in the real body of the real patient, lead to more realistic behavior, such as this can form the tissue structure. They can do like the communication with each another cell, and also react with the chemical. For example, when we treat the dark, all of the cell in the 3D structure can explode in the same condition. Anyhow, I try to put the table compare between the beginning traditional 2D cell culture with the advantage and disadvantage of each approach, from the 2D cell culture, 3D cell culture, and the advanced technique like the microfluidic, and also compare with the animal model or in vivo model. You can read by yourself, and also again compare 2D and 3D cell culture. Why the 3D cell culture is become popular in the cancer research field, because that you see is give more physiological cell-to-cell interaction and cell-to ECM, extracellular matrix interaction, that have not occurred in the monolayer of the 2D cell culture like this. But okay, and also the important point that I want to point to you is they carry out the gene expression profile mimics to original of the tumor from the patient. But compared to the 2D, they have something benefits also, because it's quite easy to handle, well established, low cost, and high throughput to screen the drug. Even you want to screen the drug 100 or 1,000 drug, the 2D cell culture will help you to finish the task. I mean the experiment in the short time, something like that. Okay, show to you again,ค่ะ. In here, 2D cell culture grow attached to the dish, plastic dish, in the monolayer, not grow up or down from the plate. But compared to the 3D structure, you see, it's called like the 3D structure, surround with something like we put the, for example, the matrix gel or the liquid matrix, something like that, to do like the support the structure of the 3D structure. And also in this structure, I mean the 3D structure is not only the cancer cell. You see, somehow we can put another molecule, another cell to join together to form the 3D and study about the cell-to-cell interaction and also cell-to-ECM interaction also. Again, as I mentioned to you that the 2D cell culture is easy to maintain, to monitor, to perform. That when you have like the order from the doctor to screen the drug more than 100 or 1000 drug, you will start with the 2D cell culture. That you see the first mention about the drug screening procedure is failed when it's want to pass through the clinical trial. What that is mean, when you use the 2D cell culture, imagine, for example, the breast cancer cell line. From now on, the immortalized breast cancer cell on the market. You search on the Google, you will got around 10 to 15 or 20 breast cancer cell line. Okay, but when you look inside triple negative breast cancer subtype or the luminal A subtype, luminal B subtype, or HER2 positive subtype, it have just only 2 or 3 or 4 cell line. Okay, but when you want to treat the drug 100 drug, for example, treat 2, just only 3 cell line with HER2 positive, you cannot say like that this drug have good for HER2 positive in the real patient in the world. Okay, so they say like many drugs fail during clinical trial. Okay, because the result from 2D sometimes it cannot apply in the real patient worldwide. Okay, so if you want to do like this, the 3D models, 3D cell culture model become the good model to test about the drug response for the doctor. Okay, and this is we call like the personalized medicine. Each patient respond one drug. Patient A respond drug cockeye, for example, or patient B respond to drug C, for example. That means this feature is quite like the big barrier between the 2D and 3D cell culture. And another thing that I can say to you that the 3D cell culture more realistic mimics the in vivo behavior. It's more aggressive with the tumor microenvironment and also the 3D cell culture. Now on is have the publication say that the 3D cell culture is carried out 99% to 100% carry out the signaling transduction or gene expression profile from the original tumor in the patient. Okay, this slide shows the structure that compare to the in the monolayer like this, but when you want to do the 3D model, you see, you have to make some structure for them. For example, we use the matrix base. For example, the matrix gel. Put the matrix gel coat to the plate like this. Okay, and let the cell form the 3D structure. Or you mix together the matrix with the cell and put the gold like this, like the dome of the matrix gel. Or you put the cancer cell with the low attachment plate. The important thing that you have to think about when you want to do the 3D cell culture is one thing, not allow the cell to attach to the plate. Okay, if it's attached to the plate, it means they can grow in the monolayer. So you can do everywhere that you can avoid the cell that attach to the dish. For example, you put the matrix gel, grow the cell in the matrix gel, or use the U-shape well like this. U-shape is have the they have the capable to avoid the cell to attach to the well. We call it low attached well. Okay, somehow they code something like the agarose or something like that that avoid the cell that attach to the dish. And you can heard about a lot of the matrix. For example, we call the matrix gel, somehow we call the liquid matrix, or somehow we call the matrix BME, something like that. BME is the basement membrane extract. It can use in the same way with the matrix gel, or you can heard about the polyethylene glycol, something like that. It can mimics the structure of the extracellular matrix to support the structure of the 3D model. Okay, and in this slide, what I want you to think about is the 2D model. You know, it's glow just only in the monolayer, so it's not glow up or down from the dish. So if you see the cell that floating up to the in the media, it means it died. Okay, it's easy to observe to maintain the cell. If you see the cell glow like the single cell up, flow up in the media, it means it's died. But in the 3D structure, the 3D structure is mean the cell come and aggregate to form the 3D structure. So you have to concern about if it's have aggregate, it's have like the layer of the cell. Okay, it's have the layer like the. From now, we can separate like the 3 layer of the cell. From outside, we call the proliferative cell, proliferative zone. In this, we call proliferate because it's export a lot of the nutrients or the oxygen. They can grow and proliferate and make the 3D spheroid or the organoid is bigger and bigger. Okay, but next inside we call the quiescent cell. It means I say like that, not die but not glow, something like that. They're waiting for something that activate them again, and they can glow and proliferate and act as the cancer cell. Okay, what we have to concern is the in the tumor core inside, because in this side they have less oxygen. It's called hypoxic condition. They have less nutrients. Somehow, you know, in this they have a lot of autophagy inside. Okay, because they have no oxygen, no nutrients, but somehow they can die by the necrosis. Okay, so again, several papers try to compare between the proliferation between 2D and 3D cell culture, but in my opinion, it's depend on the cell line, it's depend on the cell type, it's depend on each patient behavior, I mean tumor behavior. We cannot compare exactly the proliferation rate between the 2D and 3D cell culture if when they are, for example, they are the same breast cancer cell, but if when they come from different patients, they have give you the different growth rate. Okay, so in this slide, another thing to show to you that now on they have developed the scaffold like this to become the structure of the 3D cell culture. And the scaffold you see here, they have the pore, increase the ability of the media and the oxygen to pass through to the 3D structure and enhance the 3D cell culture proliferation. Okay, again, concern about 3 layer of the 3D organoid, 3D spheroid. Okay, the outside proliferative zone export a lot of oxygen and nutrients, and they can glow up and glow up. So what I want to point to you is you have to monitor organoid by, I can say by yourself, day by day, week by week, and observe the organoid from patient, each patient, individual patient, Okay, you have to do like the record form to record about the size of the organoid, how long that is grow, how long that is suitable that you can subculture the organoid and grow it again. You can have to do like that for each individual patient, I can say. And after that, if you can do like got the result that have the stable periods to culture the organoid, you can store them in the liquid nitrogen, something like that. And then after that, if the doctor want to test again, you can activate the stock organoid and test the drug with the data that you record, how it's going, how long the day that they duplicate and proliferate. You can report something like that and use the data to apply to test the drug for the doctor. Okay, so in this slide, the method that can apply to generate the 3D cell culture. The first one, they start with the hanging drop. Hanging drop is quite easy to use. Okay, it's just like you mix together the cell, the single cell with the media, and then you drop in something like the container, something like that, and then you flip it back. Okay, and the cell will accumulate in the lower, and then they come together, aggregate to form the 3D structure. It's quite easy. No need the essential or the expensive equipment. Just the media and something like the dish or something like that. Okay, and then after they form the 3D like this, you can use them like transfer them to the 96 well plate and treat the drug, something like that. And now, as I mentioned, we have low attachment plate. They do something to the plate that not allow the cell to attach. Okay, and if the cell do not attach to the plate, they will come and join to aggregate together and form the 3D structure. Sometimes we can use like the matrix gel and also the centrifugation, centrifuge to force them to make the 3D structure. Okay, and another is the magnetic. They put the magnetic together with the cell, and then they pass through the magnetic like this to force the cell to form the 3D structure. It's the same with that you use the centrifuge to force the cell to aggregate to form the 3D structure. Okay, and now have the microfluidic is quite benefit because as you see, they have line to put the cell. 3 or 4 or 5, it depends on your money can buy. Okay, but you see the central line here, it means each cell from the patient. For example, you can put the liver cell here, fibroblast here, brain, for example, put in the different line, but they share together with the same media in the central line. It means you can study individual cell line, but in the same condition of culture. From now on, this microfluidic is quite popular for study the 3D cell culture. Okay, another thing is they use the printing, bioprinting, and also use the like the hydrogel to form the structure of the 3D cell culture. Okay, now move to the model about the spheroid and also the organoid. Okay, so is it the same that the spheroid is come from the 2D cell culture? Okay, we detach it and force them to form the 3D structure. But the organoid is mean the mimics organ, form the real tissue. They come from the tissue and then they grow inside the testicle, the dish, and then they form the structure of the 3D structure of the organoid. Okay. And again, concern about the layer of the 3D structure. Okay, we can study all of the layer of the structure, even from the outside, proliferative zone, quiescent zone, and necrosis zone. It depends on your experimental design to study about using 3D cell culture. Okay, just to show to you that in the 3D cell culture, you can see a lot of layer of the cell. All of layer you can see we have cell, all of the cell. Okay, and outside you have budding, you have proliferation ring, you have nutrient, you have oxygen, you have growth factor. But inside necrosis core, quiescent core, less oxygen, less nutrients. Okay, just to concern about that before you use the organoid to test. Okay, so again, interior organization. I have already mentioned to you 3 core that we have to concern: oxygen zone, oxygen concentration, the layer of proliferation from the proliferation until the necrosis. Okay, about the pH where we have to concern, and also the organoid can contain many types of the cancer. No, sorry, many types of the cell. For example, the tumor cell, the fibroblast, the immune cell, something like that. Okay, now look inside the spheroid. That I mentioned to you, the spheroid can derive from the established monolayer. We do like the trypsin or detached cell from the monolayer, and then from the 3D structure in the low attachment plate. You see, somehow you can use the centrifuge or you can use the metric gel to force them to form the 3D structure like that. Okay, it's quite easy to do with the spheroid. Okay, we pass this. But what I want to mention to you from now on, the scientists try to report the type of the spheroid because not all the cell line that can form the structure like the 3D spheroid. Somehow they form like the round shape, somehow they form like the mass or the crab, or somehow they have something like the spindle. They have the long leg. If you see like this, elongation leg or body, something like this, can refer to the cell ability to migrate through the matrix gel. Okay, if they just go in the round shape but it's bigger, it's show the capable of the cell proliferation. But if you see the cell like the leg like this, the spin like this, it means that the cell can invade or migrate through the matrix gel. Okay, and this is, for example, in the breast cancer, you can see even they are the breast cancer cell, but if there are different subtype, for example, we know A, HER2 positive or triple negative breast cancer compared to the normal membrane breast cell, you see, they form, even they have different 2D structure, they also have different 3D structure also. If you see like this, you can, if you work closely every day with this type of cell, you just to look under the light microscope, you can guess what is come from. For example, beautiful organoid spheroid like this is come from the MCF10A. This is the normal breast cell, something like that. Anyhow, when you see like this, for example, this is my established cell line 148 breast cell line. You see, they're not round shape, but they have something like the pseudopodia. They have the leg. It means this type of the cancer patient, they have the behavior like the high invasive. Okay, and when we look at the genetic and the gene expression, we have seen a lot of high expression of gene that involved in the cell migration and cell proliferation. Okay, the application of 3D spheroid, okay, investigate cell-to-cell interaction used as the model to monitor the disease progression, used to monitor about the drug testing, something like that. You can read by yourself here. Okay, so from now on, the population is quite easy. Sorry, it's quite easy that you use the U-shape. Mix together the cell, media, and the matrix gel. And then you force, like, for example, the centrifuge, and let it go like a 3D structure like this. Okay, it takes around 4 or 5 days to make the 3D structure like this. And when you see here, it's made the pseudopodia, the leg in weight in the matrix gel. Okay, the different thing between the two, the proliferation and the migration. In the proliferation, you can see it's not have leg in weight. It means it contains the structure in the matrix gel. We use the matrix gel just only 2.5 to 4% of matrix gel. Compared to the invasive, we want to observe the capable to invade matrix. So we use absolute matrix gel. It means 100 matrix gel to form and see the leg invade through the matrix gel. It means if they have the leg can invade through the matrix gel, it have the aggressive behavior to migrate in our body. And now on, you see we have the machine to observe the migration area, something like that. Do not use like the image that I teach to you to make sure how long that is migrate, how is that proliferate, something like that. Okay, this is the image that I mentioned to you in another class, that we can use the image to measure the size of the spheroid, the size of the invasive and migration area, something like this. Observe not only the size, but also we can observe the fluorescent intensity in the spheroid. In this case, I'm saying the spheroid with the cell tracker with have how to say, which have the objective to monitor the cell proliferation when they express the reported gene or the cell tracker inside. It means the cell is living cell. But if the signal is decreased, it means the cell is gonna be die, something like that. Okay, in this paper, I try to show to you that we study in CRC coli. We treat periostin to induce the cell growth and cell invasion. And you see when we treat the periostin, this bigger cell size was observed, and also you will see pseudopodia formation in weight from the spheroid out through the matrix gel. It means that the periostin have the benefit, have the not benefit, have the role to induce cell growth and invasion. And also a little bit chip out, but just to show that this PhD student, they study about the interleukin 33 in cholangiocarcinoma cell. When she knocked down the interleukin 33 in the cell, you can see the cell is increase the proliferation and also the invasion when compared with the parental cell. Okay, they use the 3D model as the model to study. And also we can use the 3D cell culture like the establish and do the characterization of the new cell line that we have isolate from the patient. Okay, as I mentioned to you, I have already established 142 and 148. 142 is from HER2 positive cell, and 148 is from triple negative breast cancer cell. You can see the aggressive structure that they show a lot of the pseudopodia formation. They produce a lot of the leg to invade to the cell. And when we look at the phenotype, I mean the gene expression of this patient, they show a lot of molecule profile when we do like the sequencing, something like that. And we can use the 3D as the target to test the data also. Okay, and again, we can use the 3D structure, 3D model to test the T cell function. And I show to you the T cell can invade and destroy the 3D structure if when the T cell have specific target on the 3D model that we form. Okay, and also we can mix together not only the cancer cell, but also we mix together with the cancer-associated fibroblasts to form the 3D structure like this. We label the cancer cell with the green color and the cancer-associated fibroblasts in the red color and see how they join together and form the 3D structure. And also we can show that the cancer-associated fibroblasts can protect the cancer cell from the T cell killing. When compared with the cancer cell alone, you can see the cancer cell alone respond to the T cell killing around 80 or 90%. But when they have the cancer-associated fibroblasts together, it seems like the cancer-associated fibroblasts can protect the killing from the T cell to the cancer cell. Okay, we can show like that. And also I pass through the video. And just to show to you now on, we have a lot of publication about the 3D organoid. And you see that organoid is come from mimics or mini organ. From now on, they have several reports about the tissue that can derive to the organoid and test in the laboratory. For example, from brain, from lung, from liver, something like that, breast cancer. Okay, and you see the organoid is a mini organ. It's not only the cancer patient tissue, but also the healthy people is also can give the tissue to us, and we can derive organoid compare between the patient and also the healthy donor tissue. And you can see the organoid, they carry out the structure of the origin tumor also. When you see the structure of the organoid like this, you can guess where they come from. Okay, so again, we display describe what is the 3D organoid. Okay, and that what I mentioned to you, that it's not only the patient but also the healthy donor is can give the tissue to us and make the organoid to test in the laboratory also. Okay, but the important point is that the key difference between each patient. Okay, even you are breast cancer patient, but if you give us the tissue derived to organoid, your organoid will respond to the data in different way of response. Okay, and okay, I will pass. What I mentioned in this slide is that the sort of organoid can come through different way. From my expertise, I deal the 3D organoid from the primary tumor tissue. Okay, so that's why I call the 3D organoid as the PDO, patient tissue derived organoid. But the organoid can come from another sort, for example, stem cell, pluripotential stem cell, or adult stem cell, like the Ajahn Methijit or Ajahn Pan deal with the stem cell. But for me, I have expertise to derive the organoid from the primary tissue of the patient. But finally, it's the same that we try to generate the organoid to test for the patient. Okay, just to show to you is a mini organ. So it's do like the mimics the original structure from the patient. For example, intestinal, you will see they have the structure like the clip like this, and also you can see they have the budding out. They try to budding out from from this origin organoid. And as I mentioned to you, we try to set up like the standard to culture the organoid, but it depends on the type of the cancer and also depend on the each patient. You can see, for example, in this type of the cancer, you see that they use several days to form the structure like this, okay? And you have to monitor every 2 days, 3 days to observe. Is it suitable to test the drug or to study for further study or not, okay? And okay, that I mentioned to you different organ, different organelle structure. For example, the lung, you see like this, or the breast cancer or the liver cancer. You have to get different shape of the cancer organoid. Okay, and as I mentioned now on, the report about the 3D organoid come from several organs. Okay, and this is the application of the 3D organoid, test the drug, test the toxicity, the drug testing for the personalized individual patient, the drug screening, drug cancer metastasis, do like the disease model, something like that. And now on, they have tried to put the high throughput, high advanced technique. For example, they do like the biomarker detection, they do the gene profile, they do the single cell analysis to identify the gene in the organoid also, okay? And the basic concept of organoid is to mimic the structure and the function of the organ from the body, okay? And the 3D organoid that isolate form not only the stem cell but also the tissue. It can control and mimics the tumor microenvironment from the real patient. And now it have a lot wide range organ, not only listed here, but have a lot of organ that can derive to the organoid. Okay, and this I have already mentioned the medical research applied by using the 3D organoid, okay? That you can see now on, they have the omic, they have the AI, they have the genetic and sequencing, the personalized medicine, and the drug target that can test by the organoid, okay? And this slide show to you that I mentioned that you can see the organoid from each patient carry out the structure and also the gene expression profile from tissue origin. Okay, you can see the stain with the H and E, you can see is mimics to the origin tumor. You can see here, the structure is quite the same. They can say 100% the same together with the origin tumor. And also here, ER and HER2 staining show in the same pattern staining, even they are the origin tumor and also the organoid that we section to the FFPE and made the ISC staining. Okay, and this slide just show about this girl deal with the breast cancer, and she show about in one organoid contain several, I don't know.
They're quite small. We keep them under 500 microns to make them very standardized, and you can see that.
Just to show that in one organoid, she tried to show that they have contained several cells inside one organoid. Okay, and she used the fluorescent label in each cell in one organoid of the brain. Okay.
For the news.
Just like that. Okay. So come to the last. Give me just 5 or 10 minutes. I will finish about the method that can prepare 3D cell culture. That when you got the biopsy or the tumor section from the the doctor, we cut in the small piece like this. We use the enzyme and we use the temperature to isolate the cell, and then we put the we use the centrifuge, get organoid pellet, and then we put the matrix gel, and then we dome it like this. And as I mentioned to you, we dome the matrix gel that mix with the cell, and then we flip it back, flip it down. We made it around 30 minutes to let the matrix gel polymerize to hard gel, and then we flip back, and then we put the media to glow. Okay, this is the method to prepare the organoid. Okay, cut the small piece, and then we digest with the enzyme or the temperature, and then we remove the tissue, get only the single cancer cell. Somehow we can use the lysis with the red blood cell lysis buffer, and then we embed with the matrix gel, and then we observe from day one. You will see the single cell, and after that you will see the organoid grow up in the matrix gel. Okay, I will pass through here and want to show to you that we drop the matrix gel that mixed with the organoid pellet, flip it back, flip it down 30 minutes in the incubator, and then put the media. You will contain several organoids in one dome of the matrix gel. And the component list is here. You will see it's quite expensive to perform organoid experiment because they require a lot of component inside the media culture. Okay, it's quite expensive to do the organoid. And you see here, just one dome, you contain a lot of organoid. Okay, and even there are CRC, colorectal cancer model, but if they come from the different patient, you see patient number 5 and 30, you will see the different formation of the organoid going up until the 14. Okay, and you can see the different pattern grow up of the organoid. Okay, from the first one, they not grow, they not die, but they don't grow up, or they strongly grow up, grow outside, and this compared to this, they grow inside, but all of them they grow. Okay, so until now you have the AI. Now on we apply the AI, not measure by manual. You can use the AI to measure the size, count the area, count the size, and also they can monitor time by time for you and plot like the graph ready to you. You can use the AI to analyze the organoid formation for you. Okay, not use the manual image that I have already teach to you. Okay, I move past. Okay, why? Okay, so as I mentioned to you, now on they have the AI to monitor time by time, to monitor each organoid for you, and then they report time by time to you. We have the machine like this in, I'm quite not sure that some kind of you saw in Ajahn Sompunnath or Ajahn Siwanol lab, they have the machine like this, the automation machine that can see the organoid for you, monitor organoid for you, do like the media changing, do like the treat the drug for you, monitor time by time with the computer, and also capture the picture with the fluorescent for you, do the report for you in the real time. Okay, and you can see here from the monitor, starting with plating the cell, feeding the media, do the subculture for you. You can see it's grow up, the report grow up, the number and the size of the organoid, and do subculture again, grow it up again, subculture again, grow it up again. They can report like the real time and do the report for you, and you can report like the stable time, stable monitor to report this type, this patient organoid for this patient have the periods of the culture I can say, for example, 5 days for one doping time, something like that. And also you can see total area and organoid number they can report for one machine. Grow up subculture, grow up subculture, grow up subculture can do it for you. Okay. And also you can apply the organoid study about the gene expression. You can select the marker that you want for in different patient. Okay, for example, the basal marker is the cytokeratin. Is the basis of the epithelium cell marker. Okay, and you can apply another marker that you want. For example, CD24 and CD44 is a marker for the stem cell. You can also apply and study in your organoid. And also, you can see here, even they are in the same organoid, but they express different formation and also different gene expression in this. You see, it's express this marker. That means they already want to move out. They want to budding out from this organoid. Okay, and use the organoid to trace the drug. Okay, test the drug in different concentration and plot the graph to report that they respond to the drug, rapamycin and also the doxorubicin. And as I mentioned to you that now on the AI can monitor organoid by time for you. Okay, I will move and I will finish. Okay. Okay, not only treat the drug, monitor morphology, and also you can do like the stain the protein of interest in the real time, okay, by using the organoid. Okay, and this is the machine to observe. This is the one well, each well. I think this is 96 well plate or maybe 300 well plate that observe the organoid inside, and they can report the size and the number of organoid in one well for you. Okay, and now they have the AI, they have the single cell analysis and omic analysis to study about the gene profile in one organoid, compare each condition to you. Okay, and this is my favorite paper that I love to read because they try to use the organoid join together with the T cell. You can see the line, different line color is one T cell. And after they put together the T cell and the organoid, they can classify the T cell, starting with the dying, the lazy T cell, or the engager, super engager T cell to kill the cancer cell. They monitor T cell, one T cell, do like monitor they move, they join to the organoid and then they move out, the joy and they move out, the joy and they move out. And this paper they try to classify the T cell like this, lazy T cell or super engager T cell to kill the cancer cell. And they can show like the CD4 T cell and CD8 T cell. They can show like the CD4 T cell, they love to join to the cancer cell, but they do nothing to the cancer cell. They come again and they will help to kill the cancer cell. Even compared to the CD8 T cell, you see, it come at the first time and they destroy the cancer cell. I mean the organoid cancer cell. They can show the behavior and the function of the T cell, and they classify the cancer the T cell in 9 group like this. Okay, and okay, this is my point. Okay, I will end up with this future area of the organoid application. As I mentioned to you, now on we all of us try to make like the conference to join and include all of research about the organoid, and we try to set the standard to make like the method to produce the organoid in the same way, in the same standard. But it's hard to do like that because it's not the same in different patients, in different cancer models. So now that is the big challenge for us to like set the standard for do the same way, get the same result, success result for the organoid. And another thing that we want to do is the large scale production of the organoid. Okay, from now we can do like the multi organoid system. For example, we do like the one patient with the multi-drug testing, or we use like the microfluidic like this. We try to build a machine that can put organoid from different organ from the same patient and test like the same condition, same treatment, and the same condition, medium, something like that. But organoid is come from different organ, but joined together in the same situation, something like that. Okay, so I will end up. Sorry for my late. Any question from you guys?มีคำถามไหมคะภาษาไทยก็ได้ถ้าไม่มีไหมคะมิวมีแจ้งไหมคะฮัลโหลค่ะ
So organoid seems to be a better option to like study the cancer nature. So I wonder what are the choices to consider when consider to use organoid and spheroid ครับ So what are the key like what are the key factors that need to consider in order to like decide whether to use organoid or spheroid?
Okay, that and as I mentioned to you, and normally the spheroid is come from the the commercial cell line that you have in the lab. The commercial cell line is mean the cell that you have grow in the 2D structure already. You just to force them to make like the 3D structure to mimic the tumor in the real patient. But the organoid, patient-derived organoid, is the organoid that come from the 3D structure that come from the real tissue from the patient. Okay, so it's different approach. Even your is this breast spheroid and breast cancer organoid, but as I mentioned, the spheroid that come from the 2D structure is not carry out 100% gene expression from the real patient. It's just like the representative cell line. Okay.
Oh, okay ครับ thank you.
Yes. If no any question, พี่หมิว announce something ไหมคะ about tomorrow course? หรือว่าพี่หมิวได้แจ้งแล้วไหมคะว่าพรุ่งนี้ tomorrow อาจารย์ปัน course has cancelled? ใช่ไหมคะ Okay. Okay, so tomorrow you have just only the lecture from Joanne, okay, at 5 p.m. Okay ค่ะ Joanne is English is quite good and hard to understand, so you have to learn the Scottish English. Okay, so if no any question, I close this section นะคะ